Welcome back to HIV RNA Test Guide Podcast.
And hi, everyone.
You know, an effective HIV vaccine,
Yeah.
it always felt like kind of a distant dream, right?
Right.
Almost impossible,
Yeah.
because the virus is so good at mutating,
Absolutely.
constantly changing,
Yeah.
outsmarting us.
Right.
Every time we feel like we're close
Yeah.
to cornering it.
It's tough.
But
Yeah.
the research that you sent me
Uh-huh.
has me feeling optimistic.
Yeah.
Scientists are testing some really innovative HIV vaccine approaches
Mm-hmm.
that could provide broad protection against these strains.
Yeah.
And the results are promising.
It's really, really exciting.
But what's so groundbreaking about this research?
Well, I think what's really different about this
Yeah.
is it's fundamentally different from what we've tried in the past.
Okay.
Past vaccines focused on targeting specific HIV strains.
Right.
And that was like trying to hit a moving target.
Yeah.
HIV, it's a master at dodging those bullets, right?
It mutates so fast that those vaccines became ineffective.
So it's like we were trying to build a fence to contain a shapeshifter.
Yeah, exactly.
Like by the time you think you've got it tracked,
Right.
it's morphed into something else
Look at
and
the
sort of
stuff.
slipped through the cracks.
Exactly, yeah.
And that's why this research is so exciting.
Yeah.
Instead of chasing after individual strains,
Mm.
scientists are now focusing on training the immune system
Yes.
to recognize and attack a broader range of HIV variants,
Right.
regardless of how the virus tries to mutate.
It's a big shift in our thinking.
Okay, how are they doing that?
Well, the key lies.
What's the secret weapon?
The key weapon here are broadly neutralizing antibodies,
Okay,
or bnAbs.
bnAbs.
bnAbs. Think of them as like the elite special forces of your immune system.
I like that.
They're highly trained to recognize and neutralize a wide variety of HIV strains,
Okay.
effectively taking down multiple enemies with one shot.
Now you're speaking my language. Right.
Yeah. So our bodies can already make these bnAbs,
Mhm.
but they're really rare.
Right.
It's like having this team of expert snipers, but only a handful of them are actually deployed.
Right. So the challenge is figuring out how to get our bodies
Exactly.
to make more of these bnAbs.
Yeah.
And that's where this germline targeting strategy comes in.
That's right.
Germline targeting.
Germline targeting.
That sounds pretty complex.
It is a little bit complex.
Can you break it down for me?
Yeah, so imagine you have a recruit with the potential to become
Okay.
like a top-tier special forces soldier,
Mm-hmm.
but they need the right training, right?
Right.
So that's essentially what scientists are doing with these bnAbs.
Okay.
They're developing special molecules, we call them immunogens,
Okay.
to prime the immune system.
Right.
It's essentially providing those recruits with their basic training.
Got it. So it's like boot camp for the immune system,
Exactly.
prepping
Right.
those potential bnAbs for battle.
Yeah.
Uh,
And this priming process creates precursor B cells.
Okay.
Those are the recruits that have the potential to evolve
Right.
into those really powerful bnAbs.
Okay.
Ones like VRC01,
DRC01,
BG18.
BG18.
Yeah.
So are those like the star players on this special forces team?
You could say that.
Okay.
VRC01, for example, has been shown to neutralize over 90%
Oh,
of diverse HIV strains.
that's a huge number.
Huge number,
So, okay, so we've got this basic training, right?
right?
The priming.
The priming.
What comes next?
Then comes the boosting phase.
Okay.
So you've got the recruits who've completed basic training.
Mm-hmm.
Now we give them the specialized training to develop specific skills, right?
Oh, yeah.
So scientists design booster immunogens that further refine
Okay.
those precursor B cells,
Right.
pushing them to become those mature
Okay.
HIV-fighting bnAbs.
Got it. So it's a two-step process, prime and boost.
Prime boost.
And you said they've been testing this in preclinical trials, right?
Right.
That's right.
So what have they found?
So they've actually conducted 4 preclinical trials,
Okay.
both in mice and in macaques. And the results so far have been really promising.
What have they found specifically?
Well, in one trial,
Yeah.
researchers were able to successfully prime those rare
Okay.
BG18 precursors in a wild-type animal model.
Wait, wild type.
Wild type.
So these are animals with normal immune systems.
Yes, normal immune systems.
Not genetically modified or anything like that.
Exactly.
That's really impressive.
Yeah.
And to make sure that they were actually priming the right kind of cells.
Mm-hmm.
They used cryo-EM structural analysis.
Yes.
Okay.
Which is like,
Cryo-EM, that's a mouthful.
it is a mouthful.
What is that?
It's like a super high-powered microscope.
Okay.
It lets them visualize the structure of molecules in incredible detail.
So they used this cryo-EM to essentially confirm that they were creating the right kind of recruits.
Exactly.
Yeah.
It provides visual confirmation
One ad.
that the priming process was working.
Okay, so 1 trial down, 3 to go.
3 to go.
What about the others?
Well, in another trial,
Yeah.
they took things a step further.
Okay,
They incorporated RNA technology.
hold on.
Yeah.
RNA technology.
RNA technology.
I feel like there was a lot of hesitancy around that with the COVID vaccines.
Understandably.
How is this different?
Well, so RNA technology,
Okay.
it's a powerful tool.
Okay.
It has a lot of potential applications.
Mm-hmm.
In this case, they used it
Okay.
to create this prime boost regimen
Right.
that specifically targeted those B cells
Okay.
and pushed them towards becoming those mature bnAbs.
So they're essentially using RNA to deliver instructions
Basically.
to the immune system saying, hey, we need more of these bnAbs special forces.
That's a great way to put it.
Okay. And what did they find in that trial?
They found,
It worked.
yeah, the results from this trial were also really encouraging.
Okay.
They were able to effectively prime and boost those B cells,
Mm-hmm.
showing the real potential of RNA technology in this approach.
So 2 down, 2 to go.
2 to go.
What about the third one?
So the third trial
Okay.
focused on the VRC001 class
Okay.
of bnAbs.
The RC1 class, right?
Right, and they primed a mouse model
Okay.
with the same immunogen that was used in a 2022 human clinical trial.
Wait. A human trial?
Yeah.
They're already testing this in humans.
Yeah, they're moving fast.
Wow, that's incredible.
Which is a good sign.
Yeah.
And the data from that human trial really informed this preclinical work.
Okay.
They basically took what they learned from the humans
Oh.
and used it to refine
Yes.
their approach in the mice.
That's a smart strategy.
Yeah.
So they primed the immune system
Right.
to create these VRC01 precursors.
Yes,
Did they use a booster immunogen?
they did.
Okay.
They developed a brand new booster immunogen specifically designed to enhance that immune response
Ah.
and to push those VRC01 class B cells closer to becoming those mature bnAbs.
So it's like taking that specialized training to
Right.
the next level?
Yeah.
What did they find? Did it work?
It did work.
Okay.
They were able to develop a really successful prime boost regimen
Okay.
that showed real progress towards those mature VRC01 class bnAbs.
Okay, 3 down, 1 to go.
1 to go.
What was the focus of the final preclinical study?
The fourth trial was all about really understanding the fine details
Okay.
of that immune response.
So they were zooming in,
Yes,
getting a close-up view of the inner workings of the immune system.
exactly.
So-
What did they find?
So they focused on something called germinal centers,
Okay.
which are these specialized training grounds for B cells.
Okay.
They're within the lymph nodes.
So it's like the ultimate boot camp.
Yeah.
Where those B cells go through rigorous training
Yes.
to become expert virus fighters.
It's a really great analogy. And by studying those germinal centers,
Right.
researchers could see
Yeah, I could see.
how this prime boost regimen was actually affecting
Yeah.
B cell activity.
Exactly.
What did they find?
Well, what they observed was that the prime boost approach actually increased the activity of those precursor B cells in the germinal center.
So it was like supercharging their training,
Yeah, exactly.
making them work even harder
Yes.
to become effective HIV fighters.
It's a really promising finding.
Okay, so we've got 4 preclinical trials, all showing really positive results.
All really promising.
It sounds like there's a lot of momentum
Yeah,
behind this germline targeting strategy.
absolutely.
What's the next step?
Well, the next step is to take all this data
Where does this go from here?
and translate it into human trials.
So from mice and macaques to
Right.
humans,
Right.
are there any human trials in the works?
Yes, there are.
Really?
And this is where things get even more exciting.
Yeah.
There are several ongoing phase 1 clinical trials
Wow.
evaluating these vaccine approaches in humans.
They're already testing this in humans.
Yeah, they're moving quickly,
That's incredible.
which I think reflects the confidence the researchers have in this approach.
Tell me more about these human trials.
Yeah.
What are they looking at?
Well, there are a few different trials underway.
Okay.
2 of them, they're called IAVI G002
Okay.
and G003.
G002 and G003.
They're evaluating vaccines designed to prime and boost
Okay.
those VRC01 class antibodies.
VRC01, that's the one that can neutralize over 90%
That's right.
of HIV strains.
Exactly.
So these trials are particularly exciting.
Very exciting.
What about the other trials? Are they focused on VRC01 as well?
There's another trial
Okay.
called HVTN-144.
HBTN 144.
Yeah.
And that one,
That one's evaluating a vaccine designed to prime those BG18 class responses.
okay, so we've got trials looking at both VRC01
Yeah,
and BG18.
Yeah.
And these are all phase 1 trials, right?
that's right.
Just to remind our listeners,
Yes.
what does that mean?
So phase 1 trials are really the first stage of
Sure.
testing a new drug or vaccine in humans.
So it's all about safety first,
Exactly.
making sure that these vaccines
Yeah.
are safe for people.
Yes, safety first.
Okay.
And they'll be monitoring to see if the vaccines trigger the right immune responses.
Okay, so it's still early days.
Very early.
But the fact that they're already testing this in humans,
Yeah.
combined with those positive results from the preclinical trials,
Right.
it's really promising.
It's very promising.
What do you think? What's the overall significance?
Well, I think that this research really represents a paradigm shift
Yeah.
in how we think about and
Yeah.
approach an HIV vaccine.
Okay.
For decades, we've been trying to hit that moving target,
Yeah.
but now we're taking a completely different approach.
Right.
We're basically teaching the immune system to adapt and evolve along with the virus.
So it's like we're finally fighting fire with fire.
Yes.
We're giving the immune system
Yes.
the tools it needs to outsmart
Absolutely.
this really clever virus.
It's a very clever virus.
And we're seeing success in both preclinical and early clinical trials.
Yeah,
That's incredibly encouraging.
it is.
It gives me
Yeah.
real hope
Yeah, I agree.
that we might actually be on the right track
Mm-hmm.
towards developing a safe and effective
Fasted.
HIV vaccine.
It would be incredible.
It would be incredible.
Yeah.
And I know our listeners are probably eager to hear more about
Sure.
how these trials are progressing.
Absolutely.
Is there anything you can tell us about?
Well, it's important to remember that we're still in the very early stages of this research.
Right, so it's going to be a while
It's going to be a while
before we know for sure
before we know for sure.
if these vaccines are going to be effective in humans, right?
It takes time to rigorously evaluate new vaccines and therapies.
Right.
But I think the fact that we're seeing such positive results in the preclinical trials,
Yeah.
it's a really, really good sign.
It definitely gives us reason to be hopeful.
Yes.
So what's the next big milestone
Well, the next big milestone
you should be looking out for in this research?
will be the results of these ongoing phase 1 clinical trials.
Okay.
Once those results are released,
Right.
we'll have a much better understanding of how these vaccines are performing in humans
Right.
and what the next steps might be.
This is truly an exciting time in HIV vaccine development.
It really is.
It feels like we're finally making some real progress
Yeah.
after decades of setbacks.
It's been a long road,
It has.
but it's an exciting time.
This research has the potential to be
Oh, absolutely.
truly transformative
Yeah.
for millions of people around the world.
Around the world.
It could really change the
Yeah.
face of HIV prevention and treatment
It could change everything.
as we know it. And it's just incredible to think that we might one day have a vaccine that can protect people
Yeah.
from this devastating virus.
It's an incredible thought.
It is.
Yeah. This is a remarkable time to
Yeah.
be involved in HIV research.
It is.
The scientific community is making real strides, and I'm very hopeful that we'll see even more progress in the years to come.
Well, we've covered a lot of ground in this first part
Yeah, we have.
of our deep dive.
Absolutely.
We delved into the challenges of HIV vaccine development. We explored this really incredible new approach
Right.
involving bnAbs and the germline targeting strategy.
Yeah.
We discussed the exciting progress being made in both preclinical and clinical trials.
Absolutely.
But before we wrap up this part
Yeah.
of our deep dive, I want to leave our listeners with something to think about.
Okay.
We've talked about how this new approach
Yeah.
is showing great promise.
Right.
But what other challenges do you think
Oh, that's a great question.
researchers might need to overcome to develop a truly safe and effective HIV vaccine?
Well, I think you're right. While these findings are incredibly exciting,
Yeah,
the path to a truly effective HIV vaccine is still a complex one.
yeah.
There are a lot of hurdles that researchers need to overcome.
For example, we've been talking about how important it is
Right.
to stimulate bnAb production.
Yeah.
But we also need to make sure that these bnAbs are durable.
Absolutely.
We need to make sure
Yes.
they persist in the body long enough to provide that lasting protection against HIV.
Yeah.
And of course, safety is paramount.
Safety first.
We need to make sure that
Yeah.
these vaccines don't cause any unintended side effects.
And we also need to remember that HIV is a very diverse virus.
Right.
You know, there are many different subtypes circulating around the world. A truly effective vaccine would need to provide protection against all of them.
Yeah, that's a big challenge.
That's a tall order.
But I have faith in the scientific community
Yeah.
to find solutions.
Me too.
This is definitely a complex challenge.
It is.
But I'm feeling optimistic
Yeah.
after learning about these new approaches.
Me too, I think.
It feels like we're finally moving in the right direction.
I agree. It's a really, really remarkable time
It is.
to be involved in this research, and I can't wait to see what the future holds.
Well, that brings us to the end of part 1
Yeah.
of our deep dive into these innovative HIV vaccine approaches.
Okay.
We'll be back soon with part 2,
Sounds good.
where we'll delve even deeper into the science
Okay.
and explore the potential global impact
Yeah.
of these really groundbreaking discoveries.
I'm looking forward to it.
Stay tuned.
All right. As we dig deeper into all of this science,
Yeah.
it's easy to get lost, right? In all the technical details. But it's really important to remember that behind all of the complex terminology and all of the scientific breakthroughs, there are real people whose lives are affected
Absolutely.
by this virus.
The human impact of this research
Absolutely.
can't be overstated.
It can't.
HIV is a global health crisis.
Yeah.
It's touched countless lives. And it's crucial that we keep those individuals and those communities at the forefront of our minds
Absolutely.
as we're exploring these advancements.
For decades, people living with HIV and those at risk have been waiting for a safe and effective vaccine.
A vaccine that could finally offer them
Yeah.
protection and hope for the future.
They've endured the stigma.
Yeah.
They've faced discrimination, and they've had to navigate a really complex and often challenging healthcare landscape. Their stories are really powerful reminders of why this research is so important.
It is.
And why we need to keep pushing forward to find a solution.
And their resilience and their advocacy
Absolutely.
has really played a vital role
It has.
in driving this research forward.
It really has.
They've been instrumental in raising awareness,
Yes.
advocating for more funding,
Right.
participating in trials,
Absolutely.
and really shaping the research agenda to make sure that it aligns with the needs of those most affected by HIV.
Exactly.
Yeah.
Community engagement is essential for ethical and effective research.
Right.
And it's really heartening to see
Yeah.
how researchers are increasingly prioritizing this.
So what does that look like in practice?
So in practice,
How are researchers actually engaging with communities?
okay, so
By making sure that their voices are heard.
it takes many forms.
Okay.
For example, researchers are increasingly involving community advisory boards
Okay.
in the design and implementation of clinical trials.
Got it.
These boards provide really valuable insight into the cultural and social and ethical considerations that need to be taken into account when you're conducting trials.
Okay.
So it's about making sure that those trials are culturally sensitive, that they're accessible and responsive to the needs of diverse communities.
That's really important.
Yeah.
It's about making sure that the research is inclusive and that it actually benefits the people it's intended to help.
Exactly.
What else are researchers doing?
Well, education and outreach are also really key.
Okay.
Researchers need to be able to communicate clearly and effectively about all this complex science. So that means dispelling myths and misconceptions about HIV vaccines and addressing any concerns that community members might have.
So it's about fostering a dialogue,
It is,
listening to people's questions and anxieties, and building trust through transparency
Transparency is very important.
and open communication.
Yes.
And it's not just about sharing information one way.
It's not.
Right. It's about
Two-way street.
creating a two-way street
Yeah.
where researchers are actively listening
Actively listening.
to the lived experiences of those impacted by HIV.
And incorporating that feedback.
And then incorporating that feedback
into the research.
Into the research process.
Exactly.
Okay.
And researchers are also involving community members in the dissemination of research findings.
Got it.
It's not just about publishing in scientific journals,
Yeah.
right? It's about sharing those findings with the community in a way that's understandable and relatable and accessible.
So it's about translating all that complex scientific jargon into everyday language that people can understand.
And people can understand.
And relate to.
Exactly. And this could involve community forums, town hall meetings, creating educational materials in multiple languages, and partnering with community-based organizations.
It sounds like researchers are really making an effort to bridge that gap between the scientific community and the communities most affected by HIV.
And this focus on community engagement is making a real difference.
Okay.
It's helping to build trust. It's ensuring that the research is relevant and responsive to community needs.
Yeah.
And ultimately, it's accelerating progress towards finding a safe and effective HIV vaccine that benefits everyone.
Absolutely.
So shifting gears a bit, I wanted to touch on something you mentioned earlier about these broadly neutralizing antibodies, the bnAbs.
Right.
You said they're like the elite special forces of the immune system.
I like that analogy.
It's a good one.
Yeah.
But I'm curious, what makes these bnAbs so special?
Yeah. What sets them apart?
Okay, so
from regular antibodies.
Broadly neutralizing antibodies are exceptional for a few key reasons.
Okay.
First, their ability to neutralize a wide range of HIV strains is remarkable.
It is.
They're not limited to targeting just a single strain or subtype of the virus. They're like master keys
I like that.
that can unlock and disable a whole range of HIV variants.
Wow.
Including those that haven't even emerged yet.
So it's like having a universal weapon against HIV.
Right.
A shield that can protect against this constantly evolving enemy.
It's a great way to put it,
Okay.
Another fascinating aspect of these bnAbs is their structure. They have unique features that allow them to bind to parts of the HIV virus that are essential for infection, but are also highly conserved. Which means they don't mutate as easily.
So it's like they figured out how to target.
It's like they've targeted
the Achilles
heel of virus, like those weak spots that are less likely to change over time.
Less likely to change, exactly.
Okay.
And this makes them particularly effective at neutralizing the virus.
This is really fascinating stuff.
Yeah, it is.
And the fact that we're now able to use these bnAbs as the basis for...
Well, as the base new vaccine strategies is a real game changer.
It's a huge step forward.
But I'm also curious, from your perspective, as an expert, what are some of the most exciting aspects?
Ooh, that's a good question.
Of these new vaccine approaches? What really gets you fired up about this research?
Oh, there are so many things.
Okay.
For one, the fact that we're finally starting to understand the intricate details of the immune response to HIV. I mean, that's a major breakthrough.
It is.
We're cracking the code of the immune system, you know?
Yeah.
Figuring out how to harness its power to fight this formidable virus.
So it's like we're opening up this whole new world of possibility.
A whole new
Not just for HIV, but
just for HIV.
For other infectious diseases as well.
For everything.
Yeah.
It's really exciting.
It is exciting.
And the fact that we can now design and engineer these immunogens that can specifically target those rare bnAb precursors. You know, that's a game changer.
It is.
We're essentially creating a roadmap for the immune system.
Guiding it. To produce the exact antibodies.
To produce exactly what we need to fight HIV.
It's like we're giving the immune system.
Yes, like we're giving it
a personalized training manual.
A training manual.
Tailored to create these elite
elite
HIV fighting forces.
I love these analogies. They're so good.
I know, right?
And the incorporation of RNA technology in some of these vaccine approaches is also tremendously exciting.
It is, yeah.
It offers this new level of precision and flexibility in vaccine design.
Yeah.
You know, it allows us to deliver genetic instructions directly to cells.
Wow.
Telling them exactly what kind of antibodies to produce.
It's like we're programming the immune system.
It's like programming it.
With a new set of instructions.
With a new set of instructions.
Yeah.
It's really incredible.
And the fact that we're already seeing these positive results. In these early clinical trials. Is really encouraging.
It's incredibly encouraging.
Yeah.
It suggests that this new approach has real potential to translate into a safe and effective vaccine for humans.
There's still a lot of work to be done.
There is.
But the progress we've made in just the past few years is remarkable.
That's so remarkable.
So you mentioned earlier that researchers use something called cryo-EM, structural analysis.
Cryo-EM.
To confirm that they were priming the right kind of B cells.
Right.
Can you tell me a little bit more about how this technique works?
So cryo-electron microscopy, or cryo-EM, is a really revolutionary imaging technique.
Okay.
It's transformed our ability to visualize
the structure of biological molecules.
Wow.
At an atomic level.
Okay.
It's like a super-powered microscope.
It's like taking a snapshot
yeah.
of these molecules frozen in time.
Frozen in time, yes.
Revealing their hidden structures and secrets.
Exactly.
Okay.
And in the context of HIV vaccine research, cryo-EM has been really instrumental in helping us understand how bnAbs interact with the HIV virus at a molecular level.
So scientists can actually see how those bnAbs latch on to the virus.
They can.
Those master keys fitting perfectly into the locks.
Urza locks.
Blocking it.
Blocking it from infecting cells.
Exactly.
And by visualizing these interactions in such detail, scientists can gain valuable insights into
how to design
even more effective bnAbs and vaccines.
Vaccines, exactly.
So they can see what works, what doesn't.
Right, they can refine their approaches based on this detailed visual information.
It's like having a blueprint.
A blueprint of the battlefield.
Allowing scientists to strategize more effectively and target their attacks with greater precision. Okay, so cryo-EM is playing this really crucial role in helping us to understand bnAbs and design more effective vaccines.
Exactly.
You also mentioned earlier that germinal centers are like these specialized training grounds for B cells.
They are,
Can you elaborate a little bit more on what happens in these germinal centers?
So germinal centers,
why they're so important.
Germinal centers are these dynamic micro environments that are within the lymph nodes and other lymphoid tissues.
Okay.
And it's where B cells go through this rigorous training and selection process to become highly specialized antibody producing powerhouses.
So it's like this intense boot camp.
Yes.
Where those B cell recruits are pushed to their limits. Learning how to become the most effective HIV fighters possible.
That's a great analogy.
So when a B cell encounters a foreign antigen, like a piece of a virus, it migrates to a germinal center.
It does,
Okay.
And once inside, the B cell rapidly divides and it mutates its antibody genes.
So it's creating this diverse pool of daughter cells. Each with slightly different antibodies.
Each one a little bit different.
It sounds like a process of accelerated evolution.
It really is.
Happening right within our bodies.
It is.
It's really incredible with B cells constantly trying to come up.
Trying to figure it out. Right.
With the best antibody to fight the invader. And within the germinal center, only the B cells that produce the most effective antibodies survive and are allowed to mature.
It's like survival of the fittest.
Wow. So it's like this rigorous selection process, ensuring that only
the
strongest and most effective antibodies
make it out. Right. Yeah.
And the B cells that produce those highly effective antibodies, They're then released from the germinal center.
Okay.
And they go on to become plasma cells.
Plasma cells. And those are?
And those are the antibody factories of the immune system.
So germinal centers are like the breeding ground for these elite antibody producers.
It is, yeah.
The key to creating this powerful and lasting immune response.
It's really important. So in the context of HIV vaccine research, understanding what's happening in these germinal centers
is crucial. Crucial. We need to make sure that our vaccine strategies are effectively stimulating germinal center activity.
So it's not just about triggering any old immune response, right? It's about orchestrating this very specific response, guiding it in the right direction, to produce the most effective antibodies.
The most effective. And the research we've been talking about really highlights the importance of this targeted approach.
Right. We're moving beyond just simply exposing the immune system to HIV.
Right.
And hoping for the best.
And hoping for the best.
Right.
We're using our understanding of immunology to design these vaccines that can precisely manipulate
that immune response.
Steering it toward the production of these rare but incredibly powerful bnAbs.
It's a really big shift in our thinking.
It is.
And a really promising one.
It is.
It's a testament to the incredible ingenuity and creativity of scientists.
Yeah.
And it's offering new hope in the fight against HIV.
It is.
But it's also important to acknowledge that scientific breakthroughs don't just happen, in a vacuum.
Yeah.
The journey toward an effective HIV vaccine has been a long and arduous one. It's been marked by countless challenges and setbacks and unwavering determination.
It's a marathon, not a sprint.
It really is,
And these new vaccine approaches, these innovative strategies that are finally giving us reason to be optimistic are really the culmination of
years of relentless research, collaboration, and the unwavering belief that a solution is possible.
It's really inspiring.
It is inspiring.
Yeah.
And it's not just scientists who have played this critical role in this journey.
Right. Communities affected by HIV have been instrumental in pushing this field forward.
Yeah, they've shared their stories.
They have.
They've participated in trials, sometimes at great personal risk.
Yeah.
And they've helped shape the research agenda.
Absolutely.
To ensure that it aligns with their needs and priorities. Their courage and their advocacy have been invaluable.
It's really been amazing.
And their perspectives will continue to be crucial.
It will be.
As we move forward. Right.
It's about working together. Scientists and communities to find a solution.
Right. Benefits everyone.
Collaboration and partnership are essential for success, especially in a field like this.
Absolutely.
That is so complex and challenging.
It's very challenging.
So before we wrap up,
part
2 of our deep dive, I want to touch on something you mentioned earlier about the potential global impact.
It's successful.
The potential global impact is enormous.
It is.
A safe and effective HIV vaccine would be a transformative breakthrough.
It would.
It would have ripple effects felt around the world, first and foremost. It would offer individuals protection from this devastating virus.
Right. That's the ultimate goal.
It is,
To empower individuals to protect themselves and live their lives without the fear threat of this virus hanging over them. But the impact extends far beyond individual protection, right?
A successful vaccine could significantly reduce the incidence of HIV worldwide. And that would ease the burden on healthcare systems and allow resources to be allocated to other pressing health concerns.
So it's not just about preventing individual cases. It's about host.
It's really clear that we are on the edge of this whole new era in HIV prevention. But with all this excitement, I'm really curious to hear your thoughts on what the next few years might hold for this field.
Right.
What are some of the key milestones that we should be looking out for?
I think the next few years are going to be really crucial for HIV vaccine development.
Okay.
And there are a few key areas to watch really closely. First and foremost, those results from the ongoing phase 1 trials are going to be so valuable. They're going to give us a much clearer picture of how these vaccines actually perform in humans and whether or not they're safe and effective.
So we're all eagerly awaiting that data.
Absolutely.
And it will definitely shape the direction of future research.
Yeah.
So beyond those initial trials, what are some other developments that you're kind of anticipating?
One area that I am super excited about is this ongoing quest to develop even more potent and broadly neutralizing antibodies. Imagine bnAbs that can neutralize an even wider range of HIV strains.
Wow.
Right. Essentially, they would become like a universal shield against the virus.
Super bnAbs.
Super bnAbs,
Yeah. That would be incredible.
That would be incredible.
It's like this arms race.
It's a total arms race.
Between the virus and the immune system.
It is. And scientists are constantly working to stay one step ahead.
One step ahead.
And I think we're going to see
some
really remarkable breakthroughs in bnAb design in the coming years.
I agree.
Scientists are using all these really cutting-edge techniques like computational modeling and protein engineering to refine the structure of these bnAbs. So they're making them more effective at targeting the virus and less susceptible to those mutations.
Less susceptible,
It's like they're constantly sharpening the swords of the immune system, making them more precise and powerful.
Precise and powerful. I like that.
Another area that I'm following closely is the development of new vaccine delivery platforms. Traditionally, we relied on injections, to deliver these vaccines. But researchers are exploring a variety of these really innovative methods.
There are lots of new ideas.
Including nanoparticles and microneedles. And even oral vaccines.
Oral vaccines?
Wait, oral vaccines for HIV.
That's the goal.
That would be amazing. Imagine how much easier
it would be huge.
They would be to reach people. In resource-limited settings. Or communities where injections are stigmatized or difficult to administer.
That's a really important point.
Oral vaccines could really improve access and adherence. Make a huge difference. In global prevention efforts. And beyond improving accessibility. These new delivery platforms could also enhance the effectiveness of the vaccines themselves. Some of these platforms can actually target the vaccine directly to the cells of the immune system.
To where they need to go.
Increasing its potency. And minimizing potential side effects.
It's really exciting.
So it's like this guided missile delivering its payload right to the heart. Of the enemy. I like that. And speaking of precision, I'm also really interested in the growing field of personalized medicine. And how that might be applied to HIV vaccine development. Imagine if we could tailor vaccines to an individual's unique genetic makeup and immune system. So instead of this one-size-fits-all approach, we could create these customized vaccines.
It would be amazing.
Essentially personalized prescriptions.
I love it for protection.
Personalized prescriptions for protection. Optimized for each person.
For each person. That's incredible.
It's the future.
It is. So
we're still early in personalized medicine, but I think
it holds so much potential. It could allow us to create vaccines that are more effective, safer, and better tolerated by individuals.
And it could also help us overcome some of those challenges associated with HIV's diversity.
Absolutely.
But by tailoring those vaccines to the specific HIV strains circulating in a particular region or population, we could create these more targeted and effective prevention strategies.
Absolutely.
Ensuring that those vaccines are optimized for the specific threats people are facing.
Exactly.
It's an exciting area of research.
It really is.
That could really revolutionize how we approach HIV vaccine development.
It's amazing.
And these are just a few of the exciting developments on the horizon.
Just a few.
It's clear that the future of HIV vaccine research is incredibly bright.
It's very bright.
But I think as we look ahead, it's also important to reflect on the journey that brought us here.
It's been a long one.
We've talked about the scientific breakthroughs, the challenges, the setbacks, and the unwavering determination of researchers and communities to find a solution to this global health crisis.
Absolutely.
It's been a long and winding road.
It really has.
Filled with both moments of hope and moments of despair.
Absolutely.
But throughout this whole journey, one thing has remained constant.
Yeah, what's that?
And that's the incredible resilience of the human spirit.
That's amazing.
Our ability to adapt and innovate and persevere in the face of these seemingly insurmountable obstacles.
It's really inspiring.
It's a testament to the power of collaboration.
Collaboration.
The unwavering belief in science. And the profound impact of human compassion.
I love that.
I think so too.
Yeah.
And as we stand on the edge of this new era in HIV prevention, it's really important to remember that this journey is not over.
It's not over.
There's still work to be done.
Lots of work to do.
Challenges to overcome.
Absolutely.
And discoveries to be made.
Yeah, lots of discoveries.
But armed with this knowledge, fueled by hope, and united in our commitment to finding a cure, I think we can move forward with confidence.
Absolutely.
Knowing that a future free from HIV is within our reach.
It is. It's within our reach.
Well, on that note, I want to thank you and our listeners,
of course,
for joining us on this deep dive.
Yeah, this has been fun.
Into these innovative HIV vaccine approaches.
It has been.
It's been a privilege. To share this knowledge with you.
Likewise.
And we encourage you to stay informed.
Stay informed.
Engaged.
Engaged.
And continue to support the ongoing efforts to find a cure.
Please do.
For HIV.
Yeah.
This is a fight that we can win together.
Absolutely, we can do this.
Thank you for listening.
Thanks, everyone.
To the HIV RNA Test Guide podcast.
We'll see you next time.